Welcome everybody to our new installment of the Path to Independence series. Just to remind you, this series was introduced about a year, maybe a little longer ago, with two major goals. First is to let early stage investigators in particular, but basically all DC CFAR scientists know about scientific leaders in the HIV field, and in particular those who are working together with us at GW and other DC CFAR institutions. And the second, and probably the major educational goal, is to provide early stage investigators and scientists in their early career with some clues about how to establish a prominent career in the HIV field. And it is thus my great pleasure to introduce to you a very established and prominent immunologist who works at GW at the Department of Microbiology, Dr. Imtiaz Khan. Imtiaz actually came to GW after two other institutions in the US. And before that, he received his PhD in Banaras Hindu University in India. So he first came to US to, I believe it was Dartmouth Medical School, right? Yes. Dartmouth
Yes, that's correct.
School, and there he served as an associate professor in medicine and microbiology. After that, he moved to the Department of Microbiology to New Orleans, to this University of, how is it spelled?
Louisiana State University Health Science Center.
Yes, in New Orleans. And finally, in 2006, he joined GWU. So again, I have highest respect to Imtiaz's research simply because he runs a relatively small lab, but extremely successful. He has been funded for many years by NIH grants. He publishes in the best possible journals you can imagine, such as Journal Immunology, PLOS Pathogens, Journal Clinical Investigations, Journal Experimental Medicine. For a small lab, especially in the field of immunology, which is highly competitive, and in particular competitive in relation to HIV research, which is one of the topics of Imtiaz. This is really outstanding. And therefore, I think that Imtiaz actually epitomizes a perfect scientist who actually focuses on research, leads a small but extremely productive and highly competitive lab. And I think he may be a really great role model for junior investigators, early stage investigators, and other young scientists who are building their career in HIV field. And not only HIV field, I think that it relates also to other scientific fields. So with this, it is my great pleasure to introduce to you Imtiaz. And Imtiaz, the floor is open, and we look forward to your talk. Thank you.
Am I audible to everybody? Can you guys hear me?
I think you're fine.
Okay. Thank you very much, Mike, for this wonderful introduction. To be honest with you, when Vivian approached me, it was December and it was a grant writing time. I thought it is like any other seminar. But after talking, when I read about it, it is about your path to independence. And definitely, I have a lot of stories to tell about them. And I told Mike at the very beginning, if I go too far, please stop me, and I can stop here. So though I don't work on HIV directly, but I work on Toxoplasma gondii, which in 1986, when I came to the United States, when the AIDS epidemic was very rampant, and Toxoplasma gondii was one of the major opportunistic infection for HIV-infected population. And in one of the studies conducted by whom I call as the father of Toxoplasma research in the United States, or maybe in the world, Jack Remington at Stanford, where he published that about 30% of the HIV-infected population died from reactivation of latent disease. So most of my earlier grants, even from my mentor, were basically funded by, which was called as AOIC, AIDS-associated opportunistic infections. That was the study section we went to. But things have changed now with the heart and other antiretroviral therapy. Opportunity infections have sort of, in case of the HIV, have lost that importance. So my focus of my research in my lab is the CD8 T cell memory against Toxoplasma gondii infection. And unfortunately, I don't have control with me. I'll every time I have to tell Vivian to please go to, can I go to the next slide, please, Vivian? Okay, so just I will give you a brief description of Toxoplasma gondii. It is basically the host, the cat is the primary host where the sexual cycle takes place, and they release the oocyst. And oocyst, when the animals are grazing the field, they get ingested, and even human beings who can basically, who are dealing with cats, they also can ingest the oocyst, and oocyst basically, they liberate the cysts, tachyzoids in the gut, and tachyzoids disseminate everywhere. Toxoplasma gondii basically is very promiscuous, and it can infect any cell, a nuclear cell of the body. And it is basically, tachyzoids are taken over to the far off organs like skeletal muscles and brain, and where we don't know if it is a part of the life cycle or it is part, because of the immunological pressure, they undergo stage conversion, and tachyzoids, which are here shown, they get converted to bradyzoids, and bradyzoids is sort of a chronic phase of infection because they get cyst walls secreted around them. And it was believed that they remain dormant. Now that is not the case, because we see even in immunocompetent individuals, there is slow reactivation going on, but because if their immune system is intact, that the parasitopathogen is controlled. So primary infection, when people get infected with toxoplasmosis, and most of the infection which is passed on is through the infected meat. Because the cats or the pigs or the cows who are bearing these bradyzoids in their tissues, and they get, if they are, the meat is not well cooked, people can get infected, and seroprevalence is very high, especially in France, where it is seropositive was 70%, and in this country, about it is believed that 30% of the population has been exposed to Toxoplasma gondii. And according to CDC report, it is the second most foodborne pathogen present right now. But as I said, if the infection, if the people are immunocompetent, the infection in humans is asymptomatic, and you will not even know we are infection. You may have a mild fever or a slight enlargement of lymph nodes and so on. Birth defects is a big problem. If woman has been, is not seropositive for toxoplasma, and she gets infected in the first trimester, it will result in abortion. And in second or third trimester, it will the baby will have the birth defects. And it is a big problem in HIV-infected population, especially when the CD4 T cell count comes below 200, and anybody having toxoplasmosis will result in reactivation of toxoplasma infection. And as I said earlier, in 1986, there was a study where it was found that 30% of HIV-infected population suffered from reactivation of Toxoplasma gondii. So next slide, please. So Toxoplasma gondii is unique in the sense it evokes a very strong immune response, both innate and adaptive immune response. There is a lot of NK cell proliferation, and macrophages play a very important role, especially the ones they are activated interferon gamma. Along with TNF alpha, they produce nitric oxide, and nitric oxide is parastatic. But ultimately, the protection is mediated by T cells. If you don't have T cells, that's one of the cases of in the HIV-infected population, CD4 T cell come down, and they cannot provide help to CD8 T cells, and that infection can no longer be controlled. In the IL-12 is produced in hours after Toxoplasma infection, and it reduces the generation of CD8 effectors. And CD4 T cells there's a strong Th1 kind of response, which is manifested by very high interferon gamma production, which activates macrophages. But ultimately, in the chronic infection, it is CD8 T cells which are important, and CD8 T cells, by their ability to produce interferon gamma and TNF alpha, control the infection. And then also these CD8 T cells are highly cytotoxic, and cytotoxic effect, which is mediated by perforin, is very important for the chronic infection. And there are other cytokines which I will get to play a very important role, like IL-7, IL-15, and IL-21. Now, what is my history of toxoplasmosis research, and how did I get to be independent? Next slide, please. I was in India and basically getting frustrated and looking at these papers published in Journal of Immunology and others, that why cannot I do research? And somehow I got interested in Toxoplasma gondii, and I wanted to see what kind of immune response does it generate, and which are the antigens which induce the immune, because we didn't know much about them. Parasites are very complex structures, and there are a lot of antigens, and which are the antigens which are important, and what kind of role do they play? And at that time, I was looking at the papers and trying to apply for postdoctoral fellowship and getting rejection everywhere. And I applied to Dartmouth with this guy named Lloyd Casper, who was a new assistant professor at Dartmouth Medical School. And when I wrote to him, he told me that basically you don't have much experience in immunology, because he had isolated this antigen, which is called at P30 or a SAG1. SAG1 is surface antigen, which is a major surface protein of Toxoplasma gondii. And he had found that this P30 induces a strong antibody response, and somehow there is a T cell response also. He wanted somebody to, because he was not an immunologist himself, who can look at the T cell responses generated this P30 so that he can broaden his research. So when I applied to Lloyd, he told me, you have a limited experience in immunology, and you may not be the person I'm looking for. So I took my pen, but at least you can tell me what you want to do. And then I took pen and paper. We didn't have typewriter. Computers were not there at that time. So I didn't know how to operate the typewriter. I wrote a huge letter to him, what kind of work I did do. He said that, well, this is what you want to do is part of my NIH grant. So why don't you come? And I came there. And first of all, and these are the, I was a postdoctoral fellow. And what I found, when you take a T cells from the seropositive individuals of Toxoplasma, and you give that P30 protein, they induce very strong interferon gamma. And interferon gamma is a pivotal cytokine.
Imtiaz, there is some sound defect was, probably now is better, yeah.
Yeah, you can't hear me?
Yeah, now it's good. Now it's good.
Okay.
Sorry.
Okay, so what we found was that this paper was very well received, because what they found, interferon gamma is a pivotal cytokine for Toxoplasma gondii. If you have interferon gamma knockout mice and you give them Toxoplasma, my favorite quotation is that they can smell Toxoplasma and they will die. So when we showed that, that was a sort of a first report where we showed that one major surface protein of Toxoplasma gondii can induce interferon gamma. Then what was very important was that at that time, cytotoxic T cells were identified. This I'm talking about 87, 88, but they were considered to be most important for viral and cancer infection. The first paper which was published on the role of CD8 cytotoxic T cells in a parasitic infection was by Louis Miller's lab, which was by the guy named Sanjay Kumar in I think it was in Nature. Yeah, it was in Nature and with Michael Good, that the guy from Australia. So we were the second people who showed that in this P30 can induce parasitical cytotoxic T cell splenocytes. But ours was sort of an unorthodox finding because we showed that happens in MHC class 1 unrestricted manner. And people pounced on us because they said that, oh, an MSc class 1 was at that time, structure was just came in Nature. They said, how is it possible? We have not been able to figure this out, but there were other reports that MHC, though the classical cytotoxic T cells acts in an MHC restricted manner, but you can have in certain cases, a non-restricted situation. And then we also showed that we took a seropositive individual who happened to be the chair of the neurology at Dartmouth Medical School, who was taking care of 60 cats when he was in, his name was Alex Reeves, when he was in France, and he had sky-high titer of Toxoplasma gondii antibody. And he promised me that I can give me the cells because I wanted to clone his cells, but as when you make clones, you have to have the feeder cells every week. He said he cannot give me every week, he can give me every 2 weeks. And I generated 3 CD8 T cell clones from his Alex Reeves blood and found that one of them was highly cytotoxic. So that this P30 antigen also induces cytotoxic CD8 T cells in the humans, which was one of the major findings, that cytotoxic T cells play a role not only in the mice, but also in the humans. And then we also, in the murine model, we found that this P30 also induces cytotoxic T cells, which it is not only in MHC unrestricted manner, but it can also induce cytotoxic CD8 T cells in an MHC class 1 restricted manner. And then we cloned these P30 CD8 T cells and basically found that when you adaptively transfer this clone, you can basically protect a naive mice against a fatal toxoplasma infection. So these were my publications in the postdoc. I basically we looked at P30. It's a great antigen. It induces a good CD8 T cells. These CD8 T cells are cytotoxic, and these CD8 cytotoxic T cells are not restricted just in case of the mice, but they are in the humans only. And now, at that time, the tetramer thing had come up, and Lloyd Casper, my postdoctoral mentor, was interested in looking at the epitopes of CD8 T cell, he wanted me to create a lot of clones and so on and so forth. I said, tough luck, 5 years I have given as a postdoc. Now, even if I do, this is the problem it is. When you are somewhat productive, then people want more from you. So I was smart to the extent realized that I need my own niche if I want to be an independent. If I continue work with P30, then it will be never my project. Though those days it was because Mike told me that for about the junior investigators or first time investigators, that was a very, very important issue those days. That if your project overlapped with your postdoctoral mentor, you will never get funded. You have to create your own niche. So I was, after creating my note, now I have done enough. I have spent 4 or 5 years, published about 8 to 10 papers. Now, what is left for me next? So I started playing with cytokines. Next, please. At that time, interleukins were very much in vogue. Every 3, 4 months or 5 months, you would find a new interleukin 4, 5, 6, 8, 9, 10. Now, I don't know how it is 56 or 78. I don't remember. But at that time, I basically knew about any new interleukin which is in the market. And we had the guy who discovered interleukin 2, Kendall Smith. He was a professor at Dartmouth, and we used to collaborate with him. And actually, I used to to maintain these CD8 T cell clones, he used to supply with me with interleukin 2. He was a very difficult person. He could only get along with us, not with anybody else, but with him. He was kind enough to get interleukin 2. And many of these CD8 T cell clones which I generated was because he supplied it with the interleukin 2. And what was the trend those days? We were in toxoplasmosis, we were in competition with a laboratory of Alan Shear, who had this one very productive postdoc who's now a friend, Riccardo Gazzanelli. Actually, he's one of his relatives. He has been hired by MITM. And there was a race who will get the interleukin first, because they will get a publication in the top journal. And one of the cytokines which was interleukin 12. What Riccardo did, he showed that if you give interleukin 12 to a skid mice, you can prolong without T cells, you can generate the NK cell response to the extent that they will get greater protection, which was a new, because interleukin 12 was discovered by Giorgio Trinchieri at Wistar and by Morse Gatley at Hoffman LaRoche. And at that time, Alan Shear being Alan Shear, he had contact with all those companies. Any cytokine comes in the market, he gets it first and he gets to publish in great journals. So we were at a sort of a disadvantage. And another of our competitor was, again, Chris Hunter, who's now the chair of the in the University of Pennsylvania, Microbiology University of Pennsylvania vet school. And there was a really rat race going on. But what I showed was, if you take a normal mouse and give him toxoplasma, but you deplete interleukin 12, they die like flies. And then there were a bunch of cytokines which I was interested in. What does IL-10 and IL-7 do? So IL-10 was a sort of an immunosuppressive cytokine. When we did a northern blot, we found that in the beginning of toxoplasma infection, you get high amounts of interferon gamma. But after that, sometime you get IL-10 also. When I presented this paper in one of the conferences, Riccardo Gazzanelli says he doesn't believe this, because interleukin 10 toxo is such a strong inducer of Th1 cytokine. You don't have interleukin 10. But what is surprising, this is a lesson to you. Don't listen to anybody. Trust your data. What was surprising, after 6 months, I see his paper in Journal of Immunology, and he is showing that if you deplete interleukin 10 or use IL-10 knockout mice, the toxo-infected mice die of hyperinflammatory response, meaning that you cannot have inflammatory response going on. You need these cytokines. And that's why you see IL-10 at the later part of the acute infection. And I was under this belief that basically IL-10 may be causing, does also cause the immunosuppression, because you have IL-10 and you also have T cells. If you take them at a 7 days post-infection, they don't respond well. So I wrote an R29, or toxoplasma-mediated immunosuppression, and it got funded in the first go, and I was really excited. But I did I realize that that was only $70,000 per year and for 5 years. And it is a good way of having a free postdoc for a mentor, because with $70,000, you cannot buy animal produce salve and get other reagents. My boss was happy. I thought he was happy because I got the guy. He was also happy now that he will not have to pay my salary. So that is the challenge you face. And what happened that in those days, there was RFA came out to looking at toxoplasma and the immunoparasites which cause immunosuppressive diseases. And he told me, why don't you apply for that RFA? And I did. In the end, when the RFA was with Jack Remington, we said we should not compete with each other. Let us collaborate and send an RFA, which has a great chance of who will get funded if these two big groups combine. And at the end, Lloyd came to me. He told me, why don't I become the PI of this rather than you? And then I said that when he did it, and then I said, I have to go beyond, away from the immunosuppression and basically look at something else, so that again the issue of independence come again. So what I'm trying to convey here is that it is a big struggle. It is a big struggle to find your niche, to move away as much, at least in those days. At least nowadays, well, the study section which I go to, the SRO clearly says you don't question the independence. So, but in those days, it was a big issue, and that is a challenge. Another cytokine which I was interested in, where I really contributed, was my lab contributed that role of interferon IL-15. It was a new cytokine which was very similar to IL-2, but produced by entirely, because IL-2 is produced by CD4 T cells, but IL-15 is produced by a wide variety of cells, not CD4 T cells. And our paper in 1996 we published was that IL-15 has an effect on CD8 T cells, and if acts as an adjuvant and allows the proliferation of CD8 T cells. Subsequently, in 98, 2 papers came in Immunology by Jonathan Sprent and Everell Ma. confirming, and though they took all the credit because theirs was in a much superior journal, but there are still some people like Rafi Ahmed who do admit that I was the first person, or my lab was the first to demonstrate the role of IL-15, adjuvant-like activity of IL-15. And I know in Doug Nixon and Brett Jones and everything, they had a whole grant on using IL-15 for HIV infection. But just want to tell you that it is a documented evidence that we were the first to show that role of IL-15 in CD8 T cells. Next. This is about IL-15. It's a gamma-chain cytosol. For those who are not familiar with it, it is similar to IL-2. Although it is similar to IL-2, it has distinct characteristics in vivo. It's a great supporter of NK cells and CD8 T cells. Strong potential. There are a number of clinical trials going of IL-15 and cancer immunotherapy. And with IL-7 and IL-15, they are both involved in the homeostatic proliferation of memory CD8 T cells. Next, please. And we had a bunch of papers since then. We have published 67 papers on IL-15 in the journals which have been published in Immunology, Journal of Immunology, Journal of Experimental Medicine, showing at PNAS, showing the role of IL-15, that how critical it is for maintenance of CD8 T cells. And in this one of the papers in Journal of Experimental Medicine, we showed that if you have a Toxoplasma gondii infected mice, you block the IL-15 by using soluble IL-15 receptor, which we got from Edward. We find a CD8 T cell response, memory response is totally lost. And then we also have a paper in 2006 in PNAS where we showed that IL-15 does not affect only CD4 T cells, it affects CD4 T cells. If you don't have IL-15, the CD4 T cell help to CD8 T cells will be severely compromised. So then Raj Badra, who was a graduate student later for some time, so also as a postdoc, he did some of the studies where he showed the synchronizing effect of IL-7 and IL-15, both in the development and maintenance of CD8 T cell memory. Next, please. So basically, to get my own niche, to get my own independent focus, I was interested in CD8 T cell memory. How is why, if Toxoplasma gondii, infection like Toxoplasma gondii, generates good CD8 T cell memory, why do people get reactivation, or what happened to CD8 T cell memory? At that time, Lesenwick, I think in late 19, has said that CD8 T cell memory has different subsets. There's an effector memory population, which is present goes around the tissues, and basically it is functionally better than memory cells, but it doesn't proliferate very well to antigenic stimulation, rich stimulation. And there is another subset of memory CD8 T cells, which is called as central memory cells. They usually are in the lymph nodes. They are not functionally that active, but when they get re-stimulated, they proliferate rapidly, they produce a lot of cytokine, and they are very important for long-term memory. But this thing has expanded much more. Now we have stem-like memory cells, which not only produces effector memory cells, or effectors, they produce all the cells. You have a stem-like memory cell, they are multipotent. If they see the antigen, you can have a lot of memory cells being produced. And then there is effector cells. And then there is a resident memory cells, which is a big thing now coming up. That's the tissue-specific cells. These are the memory cells which stay in the tissues. And whether it is a lung, whether it is the skin, or whether it is the intestine, and they are there permanently. And if they receive the antigen, they proliferate rapidly. So I was interested in was, if there is, from the very beginning, I was thinking that there is some problem with the central memory cells, and how they are distinguished phenotypically. Central memory cells are CD62L positive, CCR7 positive, while the effector memory cells are CD62L negative, CCR7 negative. So this we were interested in, and actually we were meeting with failures. Raj Badra, who came with me to Washington, DC, from New Orleans as a graduate student, and we tried to see the development of central memory and effector memory cells in case of in Toxoplasma gondii infection. And it was a total mess. We couldn't get the phenotype. And Raj was smart enough at that time. What he found was that there is a paper by Rafi Ahmed, and what Rafi Ahmed shown in 2006. Can I have the next slide, please? Yeah, what Rafi Ahmad had shown that in case of 2006, that there is upregulation of PD-1 on CD8 T cells with LCMV. If you deplete PD, block PD-1, PD-L1 interaction, your CD8 T cell response is invigorated. And the mice which otherwise have a chronic LCMV infection are basically the chronic infection stops. What we found was that, or rather, we had found it earlier, but we had not taken notice of it, that if you infect a mouse with the Toxoplasma gondii infection, it gets rid of the acute infection. But after 100 or 100, between 150 days, most of these mice, they are fine, but ultimately they die. Even the acute infection has been eliminated. And what we found that the tachyzoid specific genes, it goes up, the infection reaches the chronicity, but again, at week 10 post-infections, their tachyzoids come up again, suggesting there is some kind of reactivation of infection. And if you have a bradyzoid specific genes, they are not very, at week 3, they are high, but they go down. This observation suggests to us that there is some kind of reactivation going in these mice, especially in the C57 black 6 mice, which have which get toxoplasma and encephalitis. Can I have the next slide, please? And what Raj found that there is basically increase in PD1 in case of in a lot of tissue in the spleen, liver, as the infection proceeds towards the chronicity. And you have a huge expression of spleen, this PD1 in the CD8 T cells in the spleen, liver, and the brain. So there is, because PD1 is as everybody knows, it causes exhaustion. These PD1, LAG3, and other things, they are inhibitory receptors which are responsible in the dysfunctionality of CD8 to CD4 T cell. And what we found in case of the CD8 T cell in the brain, as the infection produces 7 week, their functionality based on the granzyme B or the interferon gamma, it comes down in both in the spleen or brain. What he is showing that CD8 T cells during chronic infection lose their functionality due to probably due to increased expression of PD1. So there is some kind of CD8 T cell exhaustion, and which compromises the memory CD8 T cell response. Can I have the next slide, please? And what we found that if you give anti-PD-L1, similar to what Ravi Ahmad did, you can basically, these animals can survive. And this is all CD8 T cell dependent, because if you give anti-PD-L1, deplete the CD8 T cells, animals will die. This anti-PD-L1 treatment will not be effective. It will be effective if the CD8 T cells are intact. You treat the mice with anti-PD-1, while anti-PD-1 treated mice will survive, while the others will die. And same is the case with the functionality. The anti-PD-L treatment have greater functionality in terms of granzyme B, which is a cytotoxic molecule, and in terms of interferon gamma production. Next, please. So we see that what is really exhaustion? Exhaustion is due to the expression of inhibitory receptors like PD1, LAG3, 2B4, and now there are so many other like TIGIT and like Vistas and so on and so forth. And what inhibitory receptors do in case of chronic infection, there's an upregulation of these molecules and their functionality is gone. If you have a highly exhausted CD8 T cells, the first thing they will lose is the production of IL-2. Their CTL response will go down, and last thing which will come down is the interferon gamma production. So if you treat them with anti-PD-1, and these highly dysfunctional cells will not have only, PD-1 will be lost, they upregulated. They will also have upregulation LAG3, CTLA4, TIM3, and they will be dysfunctional. So what is going on in what has been shown in viruses or now in cancer also, that, and what we see in Toxoplasma, the CD8 T cells have high expression of inhibitory molecules, like majority of being PD1, but there is LAG3, there is CTLA4, and what we are showing in case there's the TIM3, and these cells become exhausted. Next, please. Now, again, back to when we published a lot on this exhaustion of CD8 T cells, what are the co-stimulatory molecules involved, so on and so forth. Now, we came back to this. Now, what is the memory CD8 T cell response like in Toxoplasma gondii? And we try to use the classical central memory cell markers and effector marker like CCR7 and CD62L. And one of the students who later was a technician in the laboratory, like Raj, we couldn't find it because these markers were spread everywhere. And it was my sort of a, I was not very cautious enough because I remember a long time, 2002, when I was in New Orleans, there was this immunology meeting and I had invited few people for dinners. And one of the leading virologists at the time, David Woodland, who was at Trudeau Institute, he told me that this Lanzenbachia has showed these markers of CD60, everything classical. But I get every kind of cells. It is not such a, written in the rock that you have seen. I never paid attention. And to the credit of that graduate, technician who is now in the medical school in Caribbean, what she pulled out a paper from Richard Flavel, where she found that memory cells can be distinguished based on KLRG1 expression. KLRG1 is a marker on effectors, short-lived effector cells. The short-lived effector cells are KLRG1 positive, CD62L negative. But on the basis of KLRG1 expression, in this paper by Richard Flavel, which was in Nature Immunology, you could distinguish certain memory population. And that's what we did. Next, please. And we found that if you distinguish based on the, because for CD8 T cells, because Toxoplasma, there are no, tetramers. Because it's one of the dominant tetramers, that there is one tetramer TGD057, 1.57. So we know there are not major dominant tetramers which are known for Toxoplasma gondii. We use a surrogate marker strategy, especially CD11A and CD44 for distinguishing the activated CD8 T cells. And this is our dating strategy. And we find.
Imtiaz, we have a problem with the sound again. Try again.
Okay, can you hear me now?
Yeah.
So based on KLRG1 expression and CD62 expression, we found there are 4 subsets of CD8 T cells. One is which is expressing CD62 KLRG1 plus CD62L minus. One is KLRG minus CD62L minus, and another is KRG1 plus CD62L plus, another is KLRG1 plus CD62L minus. Next, please. And these are the population 1. Population 1 is KLRG1 plus CD62 minus. This is actually sort of a memory effector cell. That's what effector cells look like. This population 2 is KLRG minus CD62. This has not been described as yet. This is more like a effector memory cell. This is a population 3 is the classical memory. Mike, please tell me where to stop because I can, basically this is, I can cut it anytime whenever you want me to. This is the central memory type, and this is sort of a transient population. What we found, KLRG1 plus CD62L plus. So next, please. We did a UMAP, and what we found on when we overlap, we get clearly 4 different CD8 subset populations. Next. And these are the population 1, which is the affected population at week 5 post-infection. It is high, but memory CD8 T cells are completely low because the acute infection is still getting resolved. Next, please. And they have different, we looked at the memory markers. As you can see, population 3 is more like a memory cells. Because they have a high expression of CD127 and CCR7 and CD43. If you look at population 1, it is more like effector population because it doesn't have much of CD127 or CCR7 or CD43. Next. And when we did a bulk RNA analysis, these are the duplicate. We have clear-cut 4 different populations. We have gone much more with the. I'm not showing you that data. Where Magley is basically spent last 6 months in doing that, and we clear-cut are convinced that we have 4 different CD8 T cell subset. One of them is more like a effector, and other 3 are the memory subsets. Next. And then we see the common genes between, they have certain genes which are common, like with the population 4 and population 3, there are about 125 genes are common, while population 4 and 3, there are 425 are different, 1535 genes are. There are some. Population 4, what we see is more close to population 1, because if you look at it, there are a lot of 1069 genes which are common. Again, we have basically explore these different populations, and we have looked at the genes which are common and genes which are dissimilar. Next. And these have different functionality. Like effector cells proliferate very well, while population 3, which we think later it's like a stem cell like population, doesn't populate, but others proliferate fine. Effector cells at week 2, as week 2, where the infection, acute infection is still there, they proliferate a lot. But at week 5, their proliferation goes down because the infection has been resolved, and 8, again, it is down. Next slide, please. And they express, as you can see, effector cells don't express much of PD-1, but population 3 and 4 have high PD-1. Well, same is the TIGIT, same with the LAG3. And as for the, again, PD-1 expression in population 2 at different points. And what is interesting in this, the PD-1 comes as early as 2 weeks post-infection. Earlier, we had thought that it is expressed at 2 weeks, but somehow the cells get exhausted at starting 5 weeks post-infection, and at 8 weeks for post-infection, these cells are totally dysfunctional. Next. Again, as I said, that when we looked at this is a spice chart of multiple receptors, and we see that population 3 and population 4 are more exhausted. Next. Again, it is the same thing. Next, please. I don't want to go through because of time. And
Dr.
what we
Khan,
did the adaptive transfer.
Dr. Khan, sorry,
adaptive transfer study.
5-minute time check. Thank you.
Okay. I will stop here, the adaptive transfer, and then I will stop here. And what we did the adaptive transfer study where we took a mice which were CD45.1. These are congenic mice which were infected by 10 cysts, and we isolated the 4 subsets of CD8 T cells from them, and basically transferred them to the CD40 equally infected mice. We took the 4 subsets from CD4.1 and transferred them to equally infected CD45.2. Next, please. And what we found that in case of the equally infected, POP1 stayed as POP1, POP2 stayed as POP3, POP3 stays at POP1 stays at POP1, and there's no conversion to POP2, POP3, and POP4. When you transfer POP2, some of it gets converted to POP1, but otherwise it doesn't get converted to POP3, POP4. And POP3, which are the stem-like cells, stay as POP3. But what is interesting, POP4, when you transfer them, all of them get converted to POP1. They don't get converted to POP2 and POP3. And when we did a transfer to the naive mice, in this case, we took a CD4 these subsets from the CD45.1 infected mice, transfer them to naive CD45.2, and then infect them. POP1 again stays at POP1. POP2 it is now converts to POP1. POP3 also converts to POP1. And POP4, nothing stays at POP4, it gets converted to POP1. Next. So this POP3, we found that they have stem-like, because one of the characteristics of stem-like cells they are TCF1 positive, L108 positive. And amongst this population, we found that the POP3 exhibits those characteristics. So POP3 is a stem-like population. The message we got from here, POP1 are the effectors. And effectors get replenished, and they have to be replenished. And the source is POP3. And POP3 gets converted to POP4, which is sort of an intermediary stage. And POP4 finally gets POP1. It is complex, but sorry, that's the way it is. So next. So I will stop here, and now because it is almost Mike has told me 10 minutes I have to give for questioning if there are some. So basically, Immunological memory is a very complex process. There are different subsets of CD8 T cells, and it can be, and this we have looked in the spleens. We don't know what is going in the brain. And it can be context infection dependent, and it can be tissue dependent. And they interconvert. And we have to look at the we plan to do a single cell RNA and find if there are additional subsets of CD8 T cells and how do they get converted. And look at the TCR diversity in these CD8 T cells. That's what we have. Our next grant is, and if we can cover, we'll be looking at that. We will look at the targets. Which cells need to be targeted to so you can generate an effective CD8 T cell response? And what is the role of CD4 T cell subsets in inducing the which CD8 T cell subset is highly dependent on CD4 T cell help? So these are the kind of things which we are involved, and we are also looking at the CD4 T cell subset. That's a different story. There is a multiple CD4 T cell subsets, and some of them have other. So that is for some other day. So a lot of people involved from that. I don't know how many. And first person, the person who's running this project, basically running the lab, is Magli Maratul. This was this adaptive transfer studies were very cumbersome, transferring CD4 T subsets separately, sorting them, and then transferring them. Major effort. It took us 1.5 years, and COVID came in between, and flow cytometry was dysfunctional for some time, but somehow we are happy we could, and we are ready to wrap the story and submit it. Thank you very much for your attention. I hope I tried to convey whatever I wanted, but again, I said these are very complex studies. And I'm happy to.
Thank you very much, Imtiaz. That's an amazing story from research perspective in particular, and very, I think, informative for those of us who use immunology. And I believe that in HIV field, almost everybody uses immunology for their research. Let me just start the question session with the following. So you presented a really impressive path that you took at different institutions towards establishing your independence in the CD8 field and accomplishing, amazing research results. I just wonder, maybe you can summarize, what in your view were the most helpful events and maybe people, and who really helped you move along this career path and establish your independence? And what were the major obstacles that you encountered and had to overcome?
Mike, I'll be very happy to. I am glad, and thanks for asking me this question. I missed it in my talk. First thing is, I will tell, if you're talking about my suggestion for postdocs is that don't always go for big names. I have sometimes, my reason of success was my postdoc, I give him credit, and also it was a problem also later on. Because day I joined his lab, I came from India, and I had never even, infection immunity was the greatest possible journal for me. Second day, he told me that, have you ever reviewed a paper? I said, no. He said, go and review this. I have a paper, whatever. You give me your suggestions. Whenever he was grant writing, he used to make me read grants and tell me what is involved in grant writing. What are the challenges? What are the successes? I gave him full credit for that. I would give 90%. Oh, yes, I was aggressive enough to contact other people like Kendall Smith, I go my own, other people. And I was not shy. I would connect. It was a great, oh, why should I go? I was very forthright in attending them. But the training I got, though he was not a big name, but he was a young assistant professor who wanted to succeed. And he understood he's a very productive, very enthusiastic postdoc, but he trained me very well. It was not just doing experiment, discussing it, writing grants, telling me how to write a grant, and what is involved. That I will give you was of great help. Problem was that obstacle was that anything which worked, he would put his name on it. Even Alan Shear told me once, your problem is that in every name Lloyd's paper is there. People will not perceive you as independent. And like I said, I'm sure, Mike, you go to study section. Nowadays, that challenge has reduced. But when I was graduate, that was a major challenge. That how do you branch off from your mentor? If in every paper you are, they will tell tomorrow it is his work, it is your work. Like I had this collaboration with Andrew Luster, who was at Harvard Medical School on Kim. He discovered IP10. This paper was going to immunity. And Lloyd told me that he had no contribution. Why don't you put my name? There was a lot of pressure. I said, Andrew told me that I can put his name, but the question is, it will take credit from you. I said, no, I suffered a lot. He created lots of problems were there. But these were the obstacles. Biggest obstacle, and my disadvantage was that I stayed at Dartmouth for quite some time. One of the Ken Murphy's postdoc told me, you don't say 100 miles in the radius of your postdoctoral mentor is. You have to do a good job as postdoc and then basically find your own niche and be in it. And that can be challenged, especially for a person like me who stays at the same institution where you did a postdoc. That's a major challenge. But I think that has decreased to, but how much? Like you review the grant. If there is some senior university, you will think, oh, is he really, though you cannot say that, but that will long answer, but that's very short. These were the challenges. Training was actually, I give him full credit for that, for training me, way he trained me. I was coming from India. You have language issues, you have other issues. But second thing is, when it came to separate out, it was not easy. It was very painful. and people have in mind, they have to go be aggressive, find a niche, and basically, that's what it is.
Thank you. Thank you, Imtiaz. Any other questions? If not, let me ask you another one, Imtiaz. I'm sorry, I just
no, it's okay. No problem.
But I'm really interested in your career, and I just wonder if you compare those three institutions where you been and you basically kind of succeeded in all of them and published wonderful research everywhere. If you compare them, where was the easiest to advance your career and where was the hardest? I understand it was different stages
I think
in your career as well, so this also kind of affects in a sense, but just comparing.
Yeah, I'll be happy to answer that question, Mike. I love Dartmouth just because of the place. It is I used to drive 1 hour. One of the reasons I attribute my success, and this is a message for authority GW, because my lab was 1 mile away from where I work. So with a postdoctoral salary, I couldn't do anything else, just go on weekends because I didn't have that much money. And I used to go to the lab. So, and you will be surprised. My easiest was Louisiana State University Health Science Center. If you can ask Magli that, and she will say, we got best publications from there. And when I left Dartmouth, people told me, you'll go to State, you'll see what the difference is. And honestly, if I look at my career graph, Dartmouth also I published in, but all the JEMs I got in Louisiana State University. Because, and though they were not top-notch scientists there, but the question is, the environment was so good. People were very cooperative. Yeah, but that's although Dartmouth is so-called Ivy League and so on. So I must say it was fine. And maybe because at Dartmouth I had to make a transition, which was very tough. In LSU, I was independent. I didn't have any strings being pulled, no pressure. That may be another reason for that.
Okay. Thank you. Thank you. Any questions from the audience? I know Alan always makes a comment. Alan, do you want to say something?
What's it been like to work in Washington, DC for you all these years? How has that impacted your research?
Washington, D.C.?
Yeah.
Because GWU, or you're talking about Washington, D.C. is Washington, D.C. problem is that there's not a strong postdoctoral culture. And you come to the building or at 6 or 7 o'clock, it is pitch dark. And I don't blame postdocs or graduate students because they live 1.5 hours away. And I don't want to pat myself back or on my back and sound immodest. In my 4 years of postdoc, I published 12 papers. And that too in Journal of Immunology, JCI, PNS, and so forth. And the reason was not I was smarter than anybody else because I lived so close, so close. The fact is, which I have told so many times, if you have an experiment and you think that, okay, I will have to come on Sunday to do this. Oh, who wants to come on Saturday? I don't want to come on Saturday. I live 1.5 hours away. Let me postpone the experiment. I will do it on Monday. Well, I was at Dartmouth. Sunday was a Sunday. So what is the big deal? I used to even walk to my lab sometimes when I didn't have a car. So this, especially, research are done by postdocs and graduate students, everything. And if for whatever reasons there are only 5 days a week, like my Lloyd Kasper, my postdoctoral mentor used to say, that science happens when you are there or when you are not there. Happens better when you are there. So I always quote this. Problem is that I don't want to be too critical but the fact of the matter is that you come on Saturday or Sunday, there is nobody in the lab.
You think it's the cost of living that makes people live so far away?
Yes, because they cannot afford it. If you write an apartment, my God, it will cost leg and arm. And so many times we pleaded, and Mike knows it very well with the former dean or the current, that there's some kind of subsidized housing. You cannot have people go in communities. It's not. On the contrary, if they are living, there are a lot of places where they are on-campus accommodation, and postdocs come on Saturdays or they leave at 7 or 8 o'clock or 9 o'clock. Basically. That's what it is, And second thing is the core facilities. These are researchers. I told you this, the work which I did. That is a bygone era. Nowadays, research is being done by core facilities. I used to come and do an experiment every 2-3 days. After now, that doesn't happen. You do have one huge experiment. You do 14 color flow cytometry. You do My lab has been analyzing this RNA bulk analysis for last 6 months. Last 6 months, we are ready. We are just analyzing. And because we don't have the core facilities. And research has changed. My fear is, I will say, after 10-15 years, if this thing continues, all the B-grade institutions will lose their research focus because there are other people, at Berkeley. There was a lady whom I invited, Magna, who was from Berkeley, and she published a great paper. All she has done is basically given the samples, and that those are analyzed by the core, and given all the core facilities, gave her the data. So when you are going to NIH and competing with these kind of people, it's very difficult. I go to, I'm on a standing member of the study section, and I see what's coming.
Thank you for sharing your wisdom and your career with us this evening. It's been fascinating to learn from you.
My pleasure. My pleasure.
Thank you.
Thanks for Mike.
So much.
I may not have gone, Mike. My apologies. I may not have gone on track because till yesterday I was a little confused. But is it a regular seminar or
Right?
Then I changed.
You did great job, Imtiaz. We enjoyed your science.
Oh, my pleasure. I'm happy.
We enjoyed to learn your career, and I think for junior investigators and postdocs, the major kind of outcome of that for their career is spend more time in the lab. The more time you spend in the lab, the more chances you make for yourself to really produce good research, produce good papers. and eventually build a career. And it doesn't matter. I mean, yes, science changed since our time with Imtiaz many years
Oldies.
but still the rule stays the same. The more
Oh,
effort
no, I
you
agree.
put in research, the more success you get. And with that, I want to thank everybody. We're at time, even past our time. I want to thank everybody for attending. I hope you enjoyed the talk by Imtiaz, and you're welcome, I suppose, to email him with questions if any questions come. And we are looking forward to our next installment, which will be presented by Dr. Tony Fauci. That will be in about 2 weeks. Tony
Tony Fauci?
Yes.
Oh my God. You should circulate it because many people.
We will show more details in an upcoming announcement with the date.
Oh, please do. It will be a pleasure to hear. What do you think? Okay.
Great.
Great. Thank you very much.
Thank you all so much.
Thanks.
Thank you.
Thank you. Thank you, Vivian, very much. Thanks, Mike. Bye.
Thank you. Bye-bye.